And our last speaker is our own Dr. Tsai talking on translating spinal cord injury research into humans.
Thank you very much for giving me this opportunity. So I think the reality is that we all know we can make a mouse walk again, and we can make a rat walk again. But clearly, none of these therapies that we've developed in rats or mice or anything, even pigs for that matter, have actually become successful. And this is not just for spinal cord injury, it's for cancer, stroke, head injury. And just to recap, I know Michael talked about riluzole, but riluzole has been found to have a primary efficacy endpoint of change in upper extremity scores at 180 days. It was not achieved. Minocycline, the efficacy of minocycline in spinal cord injury was not established. Lithium, no improvement in motor function. We've had a whole bunch of stem cell trials. None of those have actually resulted in any significant improvement. And so what we've been focused on in my lab is looking at trying to successfully translate animal therapies to humans. So we call it STAT to humans. And so our focus is not so much on a basic mechanism or anything, but trying to figure out how to expedite any of the promising therapies to humans. So how can we improve the translation to humans? Well, one is, I think as clinicians, we need to do the correct establishment of the types of patients. So we know that we need to choose the right patient. Not all spinal cord injuries are the same, and clearly there's a spectrum. We have patients who have more sensory problems or patients who have more motor problems. Some of them are unilateral, and you have more of a one right-sided versus left-sided. And so I think for those of you who are neurosurgeons, we don't treat all head injuries the same. A subdural is different from an epidural, is different from a right-sided lesion, is different from a left-sided lesion. And so to that end, one of the things that we're working on is trying to improve our ability to choose the right patients for particular trials. So with the minocycline trial, we found that if you choose thoracic patients, you're not going to find a significant improvement. Whereas cervical lesions, if you just improve one level, you can see these kinds of improvements. We're working with machine learning to try to look at the data that we have. Our group has published a few articles now looking to see if we can try to predict patients or choose these patients properly for trials and have an idea of which patients are likely to improve naturally. So that might help end some of this debate about whether or not early versus late is important, because there are probably different types of patients in these particular trials. The other aspect of it is we want to also try to pick the right patients. So currently we have CT scans, we have MRs, and we can do a surgery. If we do an MR later on, we can see there's damage. But one of the things we really want to understand, especially if we're going to try to treat these patients in these acute clinical trials, well, we want to know what actually happens to the spinal cord in that area. Now, if you're a rat, I take your spinal cord, I give you a dye, I cut your spinal cord, and I look at it under the microscope, and clearly we can't do that in humans. But we have new tractography images where we can actually start to see these fibers, and maybe we can try to choose these particular patients. So instead of the white blob versus the black blob versus the white and the length, maybe we can actually see which fibers are there. And to that end, here's an example of a patient where, if you look here on the right-hand side, it looks like there's more, it looks like there's more stenosis on one side. So typically we say, if there's more stenosis on that side, that's the side that's actually going to be damaged. And here we found that it's not. It's actually a contra-coup injury. And we can actually look at those specific fibers. And moreover, if we do these studies, we can measure something called fractional anisotropy, which is an MRI technique. And we can start to pick off, well, maybe this person has a cortical spinal tract problem, or a dorsal column problem, or a spinal thalamic problem, which are all going to be different tracts that are going to respond differently to your particular treatment. So unlike, we don't treat every head injury the same, maybe we need to better define and understand our patient population so that we can apply these clinical trials in a well-thought, meaningful fashion. The last aspect of it is to translate from animals to humans. Now, while some of you may think that some of your fellow human beings are rats, and some of them, especially at a buffet table in Las Vegas, are pigs, we don't really know whether or not humans are relevant to pigs or rats because no one's ever studied it before. So if we're looking at stem cells, who's going to volunteer to give me your spinal cord so that I can actually take it and culture it, see if they grow the same with humans? And no one's really done that. And so we're basing all these stem cell trials on animal models, and we don't know whether or not human spinal cord, primary spinal cord cells actually respond the same. So, we do cell lines. We know cell lines are warped sensitive tissues, but we can use surgical specimens, but if you're doing a surgery, they're not normal tissues. You can do autopsy samples, but you can't do that because they're dead, and you can't do cell culture, you can't do electrophysiology. And so we want to unleash expectations. And so I have some students who went through this. We're able to get live human spinal cord. And so, no, I haven't killed anybody to get it. What we do is we get them from organ donors. And to that end, we can prove that we can grow them up in the same fashion. And with that ability, we can assess for things like whether or not they grow the same in terms of differentiation factors or proliferation factors. And so I'm going to skip through this really quickly, but one of the things that we found is that they aren't the same. So, for example, they're studying things like glial scarring. Well, you know what? When we culture them in the same culture dish, humans don't make glial scars. In fact, most of the cells don't become astrocytic. They become neuronal-like. And then we find that they respond to growth factors differently. They respond to different concentrations. We can find that they respond to inflammatory factors. So at the session before, Dr. Fessler was talking about some of the inflammatory factors. Well, you know what? They don't respond the same in rats and mice. And so the last part of it is we're looking at the genetics and see if we can tease out what genes are similar between these stem cells so that we can better target this for therapies. So, we've had failure, and, one aspect of failure is the first attempt in learning. And so hopefully we'll be able to improve the translation so that we can, 1, select our patients better, 2, develop therapies that are more relevant, and 3, use the MR techniques to properly select our patients for trials. So thank you very much.